This talk is about geometry and biological processes. I will present the results obtained jointly with my student collaborator, Sarah Lanthier. Let us discuss mathematical approaches to biological processes in human body. Many of those processes are quantifiable. Some can be described by the parameters that evolve in time according to the laws from chemistry or physics. Analysis of any data obtained from the experiments involves a lot of considerations. How close the measurements are to the true state of the system. If there is any noise, how helpful the data is to the hypothesis, and about the balance between data-based and science-informed components in the model. Typically, the structure of a model involves some input states, output states, and the function from inputs to outputs. Often, the underlying mechanism is very complex or partially unknown. The process can be partly deterministic and partly probabilistic. There is extensive literature on various mathematical models in biomedical applications. Our ultimate objective is to make some useful conclusions in the hybrid models for the processes associated with pain or pain relief. We emphasize the geometric approaches. Geometry can appear for various reasons. One is dynamics of the system and the geometry of solutions of differential equations. But there are other reasons too. I note the following references. With my other student, Asma Khalid, we are preparing a journal paper on mathematical models of pain relief that uses modifications of the Hodgkin-Huxley model. My previous journal paper in 2025 is directly related to this topic. My two other recent publications on the geometric aspects of signal propagation contribute to the mathematical methods in this direction. As an example of a reference with clinical data that fits the input-output scheme with very clear conclusions, I cite a recent paper by medical researchers investigating chronic pain. Geometric perspectives in general are important in neuroscience, imaging and diagnostics, mathematical modeling of the blood flow, designing coils for MRI scanners. Investigations of the molecular mechanism of the biological processes relies on conformational changes of proteins. The computational aspects of protein structure design was the topic of the Nobel Prize in Chemistry in 2024, awarded to Baker, Sabis, and Jumper. This goes back to the earlier Nobel Prize in Chemistry awarded to Anfitson for the study of shape of proteins. To address the use of geometric methods in other areas, I give an example of reference to a paper where the computational 3D methods are used for imaging and analyzing a specific region in the human brain. Most of that is set up in the three-dimensional space. The data for the proteins in the protein data bank includes the 3D coordinates. The mechanistic models of processes are typically based on differential equations. The cellular transitions can be modeled as 0-1 switches incorporated into the equations. This setup can be expanded to include deterministic and probabilistic components together. Hybrid computational models are physics-informed and data-based at the same time. In the analysis of datasets, the use of geometry may become more abstract or conceptual. The geometric tools are applied to understand the structure of the data and the evolution in the parameter space, not restricting the consideration only to the states of the three-dimensional system. There are a lot of techniques that implement this general idea in various specific ways, including methods that go back to the ideas proposed by Belkin and Nyogi, geometric deep learning, and other methods. In the input to output framework, we would like to look closely at the biochemical processes associated with food sensitivities in humans, cell responses to antigens or to other external stimuli that lead to the release of chemicals such as histamine or cytokines or substance P and the subsequent signal transmission through the network of nerves and human body. Such processes are related to lactose intolerance or peanut allergy. These can be models for other sensitivities such as food triggers for headaches. We regard those as hidden Markov processes. In a broad sense, our approach will align with the framework of geometric deep learning. In the language of geometric deep learning, constructions from differential geometry will yield a generalized label space. The choice of parameters describing the system is important. Of course, there is a lot of discussion around dimensionality reduction, but that aside, with too few parameters, the model may not see significant effects. However, increasing the number of parameters is not always beneficial too. For example, if the project is about improving the effectiveness of imaging equipment, then adding the participant's parking registration number in the dataset will not in any way improve the outcomes. Geometry of the process can be quite interesting. Intuitively, the total space of the process is not always a tube or a cylinder. In this picture, we can see a circle continuously evolving in time that splits into 3 circles, and then those 3 circles become 1 circle again. So our system will be a human body. An input is a chemical substance, for example, a protein. An output can be another chemical, such as, for example, histamine or a specific cytokine. Alternatively, the output could be data acquired from an imaging device such as EEG or a wearable. Since the input is a chemical element, let's attempt to encode that somehow geometrically. The simple situation is when the process is just a chemical reaction. If we want to use differential geometry, then with an atom or a molecule, we should associate a smooth manifold. A chemical reaction would be a map between smooth manifolds. In geometric information theory, it is common to realize information transfer as a map of smooth manifolds. As a reference, I mentioned Menon's paper from 2021. I also bring attention to the work done at the lab of Agnieszka Kaseska in Germany. One direction of their work is to regard cellular processes as information transfer. Let's assign to a molecule a compact surface sigma of genus G, where the positive integer G equals the sum of the atomic numbers of all the atoms in the molecule. We also equip sigma with extra structure to account for bonds and other additional information that characterizes this molecule. For example, for the atom of helium, which has atomic number 2, sigma has genus 2. For both neon atom or a molecule of water, the genus would be 10, and that's why we need additional structure on sigma, such as a vector bundle, to distinguish one from the other. Here is an example of a reaction between methane and chlorine. We can interpret this reaction as a map from an appropriately chosen vector bundle and the product of surfaces of genus 10 and 34 to the product of surfaces of genus 26 and 18. In the next example, magnesium oxidation reaction is represented as a map between vector bundles on the product of surfaces of genus 12, 12, and 16 to the product of 2 copies of a surface of genus 20. Conceptually, we are aiming to represent the states of our system by geometric objects and their evolution by geometric objects as well. We interpret the process as information transfer. In any information theoretic considerations, entropy plays an important role. Since this research is done having in mind eventual applications to pain relief in humans, I give an example of a recent publication that links entropy calculated on neurological data with chronic pain. The traditional way to calculate the information theoretic entropy is as follows. For a set of positive real numbers, P1, P2, etc., Pn, whose sum is 1, the entropy is the value of the expression P1 ln P1 plus etc. Pn ln Pn times negative 1. This readily extends to allow non-negative value instead of positive. For a fixed n, the minimum entropy is 0, and the maximum entropy is natural log of n. Physicists and statisticians sometimes prefer to divide this number by the maximum value to obtain the number which is between 0 and 1. In physics, it's called disorder. I will denote the entropy by the letter E, and disorder, which is the entropy divided by the maximum possible entropy, by the letter D. In our setting, for a molecule that is formed by R distinct atoms with atomic numbers A1, etc., AR, that appear in the molecule with multiplicities M1, etc., MR, we can define the entropy value associated with the atomic numbers, which we will call E atomic, by the following formula. For example, for the molecule of methyl chloride gas, which is a small molecule that consists only of 5 atoms, the calculation yields the D value of approximately 0.6. Proteins play a very important part in cell signaling and in many processes. For proteins, another way to calculate entropy is to use the 3D coordinates of the atoms. For example, we could do it as follows. Take the file from the protein database. Assign to each individual atom the F number, which is the sum of the absolute value of its coordinates. Then, from these F numbers, we can calculate the entropy value for the molecule, which we will call E geometric, by the following formula. We did the calculation for both, for immunoglobulin, RH2, lactase, beta-NGF, beta-TGF. Immunoglobulin and RH2 are the proteins important in the mechanisms of peanutology. Lactase is central to the mechanism of lactose intolerance. NGF-beta and beta-TGF are involved with neural functions. Because these are large molecules, several thousand atoms, composed of carbon, oxygen, nitrogen, and sulfur, that have atomic numbers 6, 8, 7, 16, it's not surprising that with this choice of parameters, the entropy is close to the maximum value, and the D values are close to 1. In future work, we plan to combine geometric techniques with hidden Markov processes to make conclusions about the operator from the space of input states to the space of output states that describes or approximates the mechanism of the process. Therapy or interventions can be analyzed as extra structure on the space of input states that changes the output in a prescribed way. If you have any questions, then please feel free to contact me by email. Thank you for your attention.